Einsteins. Today I'm going to be talking to you about meiosis. Now, what is meiosis? Basically, it's a type of cell division that produces 4 daughter cells from a single parent cell with half the number of chromosomes of the parent cell. Meiosis can be split into 2 parts, meiosis 1 and meiosis 2. First, we'll look at meiosis 1. It's where the homologous chromosome pair split into 2 homologous chromosomes. The first part of meiosis 1 is interphase. It's when the cell duplicates its chromosomes and centrioles. The next step is prophase 1. The strands of DNA, chromatin, coil up into chromosomes, which are made up of 2 sister chromatids. These 2 sister chromatids, identical copies of the same DNA, are joined together at the centromere. In synapsis, 2 homologous chromosomes, which have different DNA, one from the father, let's say the red one, and one from the mother, the blue one, find each other to make a homologous pair. The mother and father chromosomes are paired up by the same gene they are coding for. In the tetrad, the formation of the homologous pair, the place where the chromosomes cross over each other, is called the crossing point. Crossing over occurs here, which is gene shuffling and switching between the mother and father chromosomes. This crossing over is important for the offspring to have different DNA than the parent. Also, the mitotic spindle fibers begin to form from the centrioles, and the centrioles start to gravitate towards the poles of the cell. The nuclear membrane also breaks and disappears. The next step is metaphase 1. Here the tetrads line up on the central metaphase plate. The mitotic spindle is completely formed and attached to both sides of the tetrad. The next step is anaphase 1. The mitotic spindle contracts into the centrioles, pulling and separating the tetrads. The cell begins to elongate. The next step is telophase 1. The chromosomes are now at opposite ends of the cell. The cell continues to elongate, awaiting division. A nuclei and nuclear envelope begin to appear around the chromosomes. Next is cytokinesis. The cell splits into 2 daughter cells. A brief interphase happens, reforming the nuclear envelope. The next part of meiosis is meiosis 2, where the sister chromatids are split. Prophase 2 is the first part of meiosis 2. The centrioles begin to move apart and begin to form the mitotic spindle. The chromosomes begin to move towards the center of the cell. Next is metaphase 2. The sister chromatids are lined up at the metaphase plate. The mitotic spindle is fully formed and attached to either side of the chromatids. Next is anaphase 2. The sister chromatids move away from each other and detach. The chromatids begin to move towards the centrioles as the mitotic spindle begins to contract. The cell also begins to stretch and elongate. Next is telophase 2. In telophase 2, the chromatids move towards opposite ends of the cell. Nuclei and nuclear envelopes begin to form around the chromatids. The mitotic spindle disappears. The cell is ready to be divided. Last is cytokinesis 2. The cells split, and there are now 4 daughter cells created from the original parent cell. Each daughter cell has a different set of chromosomes and now ready for sexual reproduction.